Morning, everybody. Let's get started. So last time, I think we already finished the, the different stage, right? The absorptive phase and, the macronutrients, how energy fuels are utilized during the absorptive phase. So we also define the stage, the duration, right? The 2 to 3 hours after meal. This time period we consider the absorptive phase or relevant to the fast state. And we also talk about how glucose, carbohydrate, and after a regular meal, standard meal, and how glucose metabolized and utilized as energy fuel, and also we finished the proteins and fat. So, so most of the fat in the meal in the absorptive phase actually is deposited in the adipose tissue for later use, right? For use during the fasting. And also limited amount of fat that we get from the diet can go to the liver and muscle tissue for storage, just a relatively small amount compared to the adipose tissue. And the levels of fatty acids was expected to be low, right? Because we mostly utilize the carbohydrates from the diet. We have a lot of enough, the carbohydrate to meet energy needs. So most of the fatty acids will be stored in the triglyceride in the adipose tissue. So the levels of fatty acids were quite low in the circulation in the absorptive phase in fat state. And a couple of things that we already mentioned before, right? The question here, which lipoprotein transports dietary triglyceride from intestine to the adipose tissue or muscle for storage? Chylomicron, right? Remember, the chylomicron is the lipoprotein that transport the triglyceride or fatty acids from the diet to the adipose tissue. And how are the triglyceride taken up by the adipose tissue? Meaning that which enzyme that has to break down the triglyceride first in the circulation in chylomicron and VLDL? The enzyme that's secreted or synthesized by adipose tissue. Lipoprotein lipase, right? LPL. Try to remember this. Okay, now let's talk about the change in the plasma levels of the glucose, insulin, and glucagon in response to a meal or during the absorptive phase. So you see here, y-axis represent the concentration or levels of the different, hormones, insulin, glucagon, and glucose levels, and x-axis is the time after meal. So after we eat, you can see the 1 hour in the both blood glucose and insulin levels goes up and reach the peak levels, right? And about 1 hour after meal. So our blood glucose level and insulin levels goes up and reach the highest level 1 hour after meal. And then you see the glucose and both glucose and decrease and return to the levels before meal, right? Both insulin and glucose. And glucagon goes opposite way. Glucagon levels decrease, and reach the lowest level 1 hour after meal, and then return back to the levels, during the post-absorptive or the before meal time period, right? So So this just to give you some idea, how our blood glucose levels and hormones levels change during the absorption phase. And also the metabolic fuel levels also, dynamic or change during the absorption phase. So you can see very clear that glucose, we already mentioned, right? Levels goes up 1 hour after meal reach the peak level, and also decline and up 2 to 4 hours. During the 2 to 4 hours after meal, the glucose levels decrease and return to the levels before meal. And amino acid levels increase a little bit and maintain relatively high levels during the absorption phase. And triglyceride levels not changing much, but increase, right, a little bit. And lactate levels actually increase a lot, which makes sense because, during the absorption phase, our body, all the tissue cells utilize a lot of glucose, right? So although not all tissues goes partial glycolysis, but some of the tissue, for example, red blood cells and other renal medulla, the cells also utilize the glucose only for partial oxidation. So these tissues will produce a lot of lactate, and so lactate level you expect to increase after meal. And fatty acids and ketone bodies, the glycerol levels will remain very low levels, right? In the fat condition, post-absorptive phase, which makes sense, right? We don't want to utilize fatty acids or ketone bodies during the fat condition. We have glucose from the diet for energy. And now let's talk about the post-absorptive phase or fasting and how our body utilizes energies or energy fuels. First of all, the definition for the post-absorptive phase or the duration. So usually we consider the post-absorptive phase from 2 to 3 hours after meal to days of starvation, depending on when we start eat food. So and what happens to the fuel utilization during the post-absorptive phase? So there is so-called a switch in fuel utilization, a switch from the utilizing exogenous fuels to endogenous fuels, and also a switch from utilizing carbohydrate to utilizing fat as an energy, right? So during the post-absorptive phase, there is the fuel utilization switch from exogenous to endogenous, from carbohydrate to fat. So, and also you wanted to know that the Randle cycle. The Randle cycle is proposed by Philip Randle. So we named this for Philip Randle. And this actually is a series to explain the mechanism, how the fuel utilization is switched during the starvation period. And also you wanted to know that how does the body accomplish the fuel switch in terms of kind of mechanism, how our body utilize fatty acid during the starvation and not utilizing glucose, right? And vice versa, how our body only or primarily utilize glucose and not utilize much fatty acids during the fat condition, right? So I think the important factors that regulate the fuel utilization switch are the hormones, insulin, glucagon, and catecholamines, and also a very important substrate during the metabolism. Some substrates or intermediates accumulate, and this intermediate can regulate the enzyme activity in different metabolic pathways, so then control the fuel utilization by switch. So for example, malonyl-CoA and acetyl-CoA are the two important substrates that can regulate the fuel utilization switch, right? And plasma levels of glucose, insulin, and glucagon during the prolonged starvation or during the post-absorptive phase. And just opposite to the absorptive fat condition, you can see the glucose insulin levels decline, right, drop a lot during the starvation and then remain the low levels during the starvation. We know that we don't want to glucose go too low, but we have our body has a mechanism to maintain the glucose level at the low level during the starvation. Just try to meet energy fuel utilization needs for brain and other tissue cells that heavily or completely rely on the glucose for energy, right? And this always pair, the glucose insulin always pair together, both decrease. And glucagon actually sharply increase, right, during the starvation and maintain at high levels during the starvation. So now I'm going to talk about the glucose utilization, right? Now we started to put the two absorptive phase and post-absorptive phase together, and then divided this two stage into the 5 different phases. Just depend on the timing, all right? The fat and fasting duration, and we divided the whole stage into five different phases. Phase 1, 2, 3, 4, and 5. And phase 1 usually equivalent or relevant to the absorptive phase, right? 2 to 3 hours after meal, and this phase considered phase 1 and also absorptive phase. And phase starts from phase 2 to 5 is considered the post-absorptive phase and starvation, right? And this diagram I initially copied from the textbook, and just for the glucose utilization or homeostasis. And because we know that during starvation, our body also utilize fatty acid and ketone bodies, right? So fatty acid ketone bodies are also very important energy fuels. So just for to be kind of comprehensive, so I just put the 2 lines here, fatty acids and ketone bodies, and see utilization increase during relative time period. For example, fatty acids usually our body utilize more fatty acids from the after 24 hours starvation, right? And ketone bodies, and after 2 days starvation, our body produces a lot of ketone bodies, and we utilize ketone body as energy. So just give you some idea, we're not only just utilizing glucose, but we also utilize the fatty acids and ketone bodies as a whole. So for the first phase 1 or absorptive phase, the glucose that we utilize primarily from the diet, right? So the exogenous sources of glucose is the most important for the phase 1 fuel utilization. And phase 2 also consider the fasting and starvation stage, that our body start utilizing endogenous fuels. So the first endogenous fuel or stored energy that we used from glucose, right? From the glycogen. So we first start to utilizing to break down glycogen and utilize glucose from the glycogen. So glycogen is important glucose sources of glucose during the phase 2, right? Or more specifically, from the 4 to 18 hours after meal, right? During this time period, glycogen is important sources of glucose. Glycogen, if the starvation is prolonged, the glycogen starts to be depleted, right? So we will utilize all the glycogens. So try to remember the time point, that after 18 hours, that our body, the glycogen start to be depleted, right? And then the gluconeogenesis is becoming important, and particularly after the 24 hours starvation. Gluconeogenesis has become the very important glucose for the body use. So gluconeogenesis even is the only source of glucose after 24 hours, because we utilize all of the glycogen. So gluconeogenesis is only source of the glucose after 24 hours starvation, right? And also our body starts to utilize fatty acids, right, after 24 hours starvation. So we break down triglyceride and utilize fatty acids. And after 2 days starvation, then ketone bodies can be synthesized and utilized as an alternative fuels. And our body try to spare glucose and amino acids and utilize more fat and ketone bodies, right, as the starvation prolongs. All right, any questions? Right. And this table, that's just pretty much similar, the information pretty much similar to the last slide, but just in different way, we try to understand better the 5 different phase and sources of glucose and tissues and other tissues utilize how other tissues utilize the different sources of glucose during the 5 different phases, and also brain, how brain utilize glucose. So for example, phase 1 basically is the fat condition, right, or absorptive phase, and dietary carbohydrate is the main source of glucose for all the tissues. So all the tissues during the phase, absorptive phase, all the tissues utilize glucose as energy, of course, including the brain. And as phase 2, glycogen is become important sources of glucose, right? And also some relatively low levels of gluconeogenesis, right? It's going on. So mainly from glycogen, and also some glucose from gluconeogenesis are the sources of glucose during the phase 2. And all the tissue except the liver, muscle, and adipose tissue utilize glucose, but at the reduced rate, right? Because it's already during the starvation period, and brain is still utilize glucose as energy, and phase 3 now gluconeogenesis, particularly the liver gluconeogenesis, now become more important, and liver gluconeogenesis rate is increased, and glycogen becomes, because of the depleted, so right, become less important. And again, all tissues except liver, muscle, adipose tissue utilize glucose, but again, at even lower rate, right? And brain still utilize glucose. And phase 4, then gluconeogenesis become very important. So phase 2, I believe it's started from almost two days later, right? 2 days, fasting. So the gluconeogenesis is the only sources of glucose, and gluconeogenesis not only from liver but also from kidney. So kidney now has also start to make glucose and contributed to glucose. So and at phase 4, which is after 2 days starvation, and then brain red blood cell, only this tissue that heavily rely on glucose, then I utilize glucose, and brain and start to utilize ketone bodies, right? And I just want to mention here, they start from phase 3 that other tissues you also utilize fatty acids for energies, right? And at a very long-term starvation, that we still do the gluconeogenesis, right? To provide glucose to maintain low blood glucose levels, and but the brain you can see that start to utilize more ketone bodies and also try to spare glucose, so at very long-term starvation. Okay, so now let's switch to the metabolic fuel utilization switch and regulation of fuel utilization switch, right? So as I mentioned that during the post-absorptive phase or starvation, particularly 4 to 18 hours after meal during this time period, blood glucose is maintained by break down the liver glycogen, right, to try to maintain blood glucose levels, and brain utilize the half of the glucose, right? The circulating glucose, half of the circulating glucose is utilized by the brain. So brain really utilizes a lot of glucose. And then glucose utilization by the other tissues slow down and try to spare glucose for brain and other important red blood cells, other tissues. And muscle does not actually contribute to the circulating glucose, right? We talk a lot about this because of lack of glucose 6-phosphatase. So muscle just utilize glycogen, break down glycogen just for muscle self-use as the energy, but not contributing to other tissue directly. And after 18 hours, remember this time point, after 18 hours starvation and liver glycogen is depleted, right? So we don't have much glycogen left for glucose. So then we'll start rely on the gluconeogenesis. So what happens after 24 hours starvation? Because glycogen is already depleted, so gluconeogenesis now has become more important for providing glucose, right? And it is only source of glucose. And also important thing is the only substrate for gluconeogenesis during starvation are the amino acids, lactate, pyruvate, and glycerol, right? So amino acids also mean breakdown, and you utilize for the precursors of gluconeogenesis in the liver and kidney, right? Because remember the kidney also become important sources of gluconeogenesis. And kidney, we mentioned before, amino acids is the most important precursors, right, the gluconeogenesis. And here's a question. So is the acetyl-CoA, which only has 2 carbons, be able to make glucose? Did we talk about this? What is the answer for this question? Is able to make glucose? No, right? So acetyl-CoA is not able to make glucose, right? Only intermediate glucose metabolites which has three carbons, like lactate, pyruvate, right, able to serve as the precursors of gluconeogenesis. Acetyl-CoA only has two carbons, not able to be used for gluconeogenesis. And the other glycolytic tissues can continue to utilize glucose, but they do not complete oxidize glucose, right, after 24-hour starvation. Tissue oxide glucose at a low rate, but also partial oxidized glucose. Just try to conserve the lactate. The lactate can be recycled into the glucose. Remember this Cori cycles, right? So a lot of tissues just partial oxidize glucose to lactate, and lactate can be recycled to glucose through the gluconeogenesis. This is a particular pathway to gluconeogenesis, Cori cycles, right? Remember. So under the fasting conditions, and not only the muscle and red blood cells provide lactate, and the other tissue can also provide lactate, right, for gluconeogenesis. And brain is the only tissue that completely oxidize glucose, right? Okay, so gluconeogenesis can provide sort of new glucose, right? Synthesized from amino acids and for the brain. So brain at this stage is still able to complete oxidize glucose, and this glucose, the new glucose, basically provided by the gluconeogenesis, right? The gluconeogenesis from amino acids. So here's the question: where do amino acids for gluconeogenesis come from during, the after 24-hour starvation period? Which tissues is the most important tissue that provide amino acids? Remember that? Yes, muscle, right? Muscle, break down the proteins and provide amino acids for gluconeogenesis. And in addition to utilizing glucose during the after 24-hour starvation, of course, most of the tissue just do the partial oxidation of glucose, right? But fatty acids now become major fuels for many tissues, particularly for muscle. Right? Muscle utilizes a lot of fatty acids for energy. And also ketone bodies can also be synthesized in the liver, right? And utilized as an energy fuel for brain. Brain now become important site for utilizing ketone bodies and also muscle. And kidney becomes a major gluconeogenic organ, right? Because after 24 hours starvation, the liver gluconeogenesis start to decrease a little bit. So kidney now become more important gluconeogenesis. Okay, so this slide gives us some idea about the plasma concentration of energy fuels during the starvation period. You can see the ketone bodies increase significantly, right? During the very long-term starvation, and glucose drops, and amino acids levels also decrease, and the fatty acids levels increase. And lactate glycerol levels actually decrease. Why lactate and glycerol levels decrease during very long-term starvation? Remember that we think that during the starvation, fatty acids triglyceride should be break down to fatty acid glycerol, right, and released into circulation. So we expect fatty acid level should be high and glycerol level also should be high. Why? During the prolonged starvation period, glycerol level will be decreased. And same thing for the lactate, right? We expect a lot of tissue just do the partial oxidation of glucose, should produce a lot of lactate, but why it decreased? Any idea? Yes. So a lot of lactate and glycerol actually being utilized for gluconeogenesis, right, to make glucose. So that's the reason, during very long period starvation, that lactate actually start to decrease, right? The glycerol and lactate level decrease. So you probably have some idea in the fat state or in post-absorptive phase, what do you expect the levels of different energy fuels, and also during the starvation period, right? So again, glucose level definitely is increased during the fat condition, but at relatively low level of fatty acids and the ketone bodies, right? And during the fasting, you expect a high level, increased fatty acids and increase the ketone bodies, but very long, and lactate levels actually increase in the fat condition, but very long-term starvation, lactate and glycerol level actually decrease, right, due to increased gluconeogenesis. Okay, so Randle cycle. So Randle cycle is actually a metabolic process that involved the competition of glucose and fatty acids for substrate utilization during the starvation, and also this series applied to the type 2 diabetes and insulin resistance. So and again I mentioned this Randle cycle is named for Philip Randle, who described it in 1963, a very long time ago. So you can see that carbohydrate utilization and fat utilization is predominant in a different physiological state, right? For example, in the fat state, our body utilizes more fatty acid, more carbohydrate. Right? So carbohydrate utilization should be high, and fat utilization should be low in the fat condition, right? And in the fasting condition, the fat utilization should be increased and glucose utilization should be decreased. So this carbohydrate and fat utilization switch or competition is kind of happening and depend on the fat fasting condition. So the question here is how our body, when we utilize more carbohydrate, how our body, what our body does is to reduce fat utilization, and vice versa. When during the fasting condition, when we utilize a lot of fatty acids, how our body try to decrease, carbohydrate utilization. Why can we do both? Increase both carbohydrate and fatty acids utilization. Why can we do this at the same time? Why one should be up and the other one up and the other one should be down? So it's kind of mechanism how our body does this. So the Randle cycle is also called a glucose and fatty acid cycle, right? And the purpose of our body to do this is the main purpose, right? Our body to do this Randle cycle is to spare glucose, right? To utilize fat, the purpose is to spare glucose. And for example, in the fat condition, when we have a lot of glucose that we obtain from the diet, and we increase blood glucose level, which causing the increased insulin level and decreased glucagon levels, right? And remember the insulin, what insulin glucagon does, the insulin can suppress the fatty acids mobilization, triglyceride mobilization, remember, right? Insulin, one of the important insulin function is to inhibit the lipolysis, inhibits triglyceride mobilization. So we have a less fatty acids released from adipose tissue in the fat condition. So we have a less fatty acid available for utilization, right? So this is the one of the important mechanisms. So then we push our body to utilize more glucose as an energy fuel. And during the fasting condition, when we have a lower blood glucose level and lower insulin and high glucagon levels, and one of the important, this glucagon and also catecholamine, right, levels increase, and then can stimulate triglyceride breakdown, triglyceride hydrolysis, right, and so mobilize a lot of fat from the adipose tissue to increase the release of fatty acids from adipose tissue. So you have more fatty acids available for energy and for utilizations, and then then glucose utilization will be decreased. So this is just the overall picture, how this fat and carbohydrate utilization is reciprocally regulated. But we have a more detailed molecular mechanism, to in the next couple of slides, right? So this just again the overall pictures of the glucose and fatty acid cycle. In this is a circulation in plasma and muscle adipose tissue. So adipose tissue is important sources of fatty acids, right? And muscle is important tissue that utilize fatty acids. So just put these two tissues together and see how the fat utilization and glucose utilization is regulated. So for example, in the fat condition, we have a lot of glucose available, right? And we can go to the both adipose tissue and muscle and for utilization. And the glucose utilization, increased glucose utilization can inhibit triglyceride breakdown, right? And can inhibit the fatty acids release. So then can inhibit the fat utilization. And vice versa, during the fasting condition, when fatty acids being released a lot from the adipose tissue, and muscle can pick up and utilize as energy. So fat utilization increases during the fasting can inhibit glucose utilization, right? So this is balanced for the energy utilization is or switch the fat energy utilization between carbohydrate and fat is called a Renan cycle or fatty acids, glucose fatty acid cycle. All right, so here is a more detailed. I have a 2 slides and describe the specific mechanism how fatty acid utilization inhibits glucose utilization, right? So this is the first slide talking about this random cycles or fuse utilization switch during the fasting condition. So during the fasting condition, as we expect, the fatty acids level will be increased, right? And fatty acids being picked up by the cells, and for example, this is a muscle cells, right? And utilized through the beta oxidation process, right? Remember the CPT1 localized on mitochondrial membrane, and and transport long-chain fatty acid-CoA to the mitochondria for beta oxidation and generate a lot of acetyl-CoA, right? Remember the beta oxidation, fatty acid oxidation product of fatty acid oxidation is acetyl-CoA. So lots of acetyl-CoA is produced, and acetyl-CoA levels increase during the fasting condition in the skeletal muscle when fatty acids oxidation is increased. And this increased levels of acetyl-CoA can inhibit, remember this enzyme, pyruvate dehydrogenase, right? Pyruvate dehydrogenase activity can be regulated by the substrate, right? Acetyl-CoA and NADPH. You still remember this, right? So acetyl-CoA levels increase can inhibit pyruvate dehydrogenase. And this enzyme is very important for the glucose oxidation, for pyruvate being further oxidized to acetyl-CoA and enter TCA cycle, right? So if this pyruvate dehydrogenase is inhibited, then glucose cannot be further oxidized, right? So the glucose utilization will be inhibited. So this is one of the mechanism, and also acetyl-CoA enter the TCA cycles, right, and generate a citrate, the first reaction TCA cycle, right? So citrate can the concentration of the citrate can also be increased if fatty acid oxidation is increased. So citrate is also important inhibitor of the two key points, key enzymes. One is the phosphofructose kinase 1, which is a key enzyme of the the glycolysis, right? And the other inhibitor point is the GLUT4, glucose kinase to oxidize the glucose, right? So this is also the other important mechanism that can inhibit glucose utilization, right? So try to remember this process. Increase the fatty acid oxidation, increase acetyl-CoA concentration, and block the pyruvate dehydrogenase, and also increase citrate and inhibits these two key enzymes in the glycolysis pathway, right? Any questions about this process? So this sort of mechanism, for how fatty acid utilization increase during fasting and inhibits the glucose utilization, right? To spare glucose. And in the fat condition, we utilize more glucose, right? And we inhibit the fatty acid utilization. So and here's the mechanism. So in the fat condition, we have higher glucose levels and go to the glycolytic pathways, right? And also generate acetyl-CoA. And because in the fat condition, we have a high insulin level, right? So insulin can stimulate ACC. Remember ACC is the lipogenesis key enzyme. So ACC can catalyze formation of malonyl-CoA from acetyl-CoA, right? And this acetyl-CoA originates from the oxidation of glucose in the mitochondria, right? And be transported through the citrate transporter to the cytosol and converted back to acetyl-CoA, right? And again, this malonyl-CoA is very important substrate, and levels increase can inhibit CPT1 activities, right? So it inhibits fatty acid transport into mitochondria and inhibit fatty acid oxidation, right? So so again, this is the different mechanism for how glucose utilization inhibits fatty acid utilization in the fat conditions. And remember that malonyl-CoA is the most important molecules that increase during the glucose utilization. That's inhibition of CPTA activated and inhibit fatty acid transport and oxidation, right? All right, so and regulation, we already mentioned, right, the hormonal regulation and also some key enzyme that does the you know the final step of the regulation, and also transporter like glucose transporters, right? I think that we have covered this before, right? About the how fat and fasting conditions the glucose levels being maintained. So I'm not going to go this again. I mean, basically just multiple tissue involved, right? Um, uh, Pancreas, liver, muscle, adipose tissue, right? They work together to maintain the blood glucose level during the fat or fasting condition. Involve the multiple hormones like insulin, glucagon, and epinephrine, right? And it's a starvation. And here I just wanted to remind you that during the exercise, catecholamine is also the important regulators of glucose levels, right? Utilization. So during exercise, the catecholamine levels increase, and liver, muscle, and adipose tissue are the main targets of catecholamines, right? So three tissues, organs, the main targets of catecholamines. So in the adipose tissue, catecholamine can increase lipolysis, increase fatty acids release, and then can be utilized by the muscle. And in the muscle, catecholamine can increase glycogen breakdown and amino acids, protein breakdown, release fatty acids, right? So catecholamine can increase catabolism to break down glycogen and protein and in the skeletal muscle. And liver catecholamine can increase the gluconeogenesis, right, to produce the glucose. All right. Do we need a break? I guess, right? Take about like 8 minutes break, right? All right, let's get it going. So this slide tells us the insulin, how insulin glucagon regulates the hepatic glucose output. And remember, for the glucose homeostasis or for blood glucose level control, the uptake of glucose from circulation to muscle and adipose tissue and other tissue is important, right, to try to reduce the blood glucose level in a fat condition. But the glucose output when during the fasting condition, glucose can also be made in the liver and contribute the blood glucose level. So the liver is also important, the organ that can regulate the blood glucose level. So in the type 2 diabetic patient, actually for some reason that glucose output that actually increased. So that also contributed to high blood glucose level, not only reduce the uptake of glucose from, by muscle and adipose tissue, but glucose output also increased. So, so glucose output, you can see from fat fasting condition. So in fat condition, blood glucose levels increase after ingestion of carbohydrate. That leads to increased insulin and decreased glucagon. And this change of insulin to glucagon ratio then can causing some metabolic change, right, in the liver. For example, decrease the glycogen breakdown and decrease gluconeogenesis, but increase glycogen synthesis, right? So the net results of this metabolic activity by insulin and glucagon ratio increase then can lead to decrease hepatic glucose output, right? So you have a less glucose produced or released from the liver in the fat condition. And during the fasting condition, decreased glucose level and decreased insulin, increased glucagon, and decreased insulin to glucagon ratio, and then also causing the change in, different metabolic pathway of the glucose. So see that the fasting condition increased glycogen breakdown, increased gluconeogenesis, and decreased glycogen synthesis, right? So all this together and leads to increase the glucose output from the liver, so that can raise the blood glucose level. And this is the normal regulation in fat fasting condition in normal physiological function. But under the disease condition, like I said, the type 2 diabetic condition, this regulation is somehow disrupted. So diabetic patients have more glucose released from the liver. And the opposite regulation I just mentioned, the insulin glucagon, right, have opposite function in terms of the regulation of glucose metabolism. I am not going to repeat this. And we also have 2 tables and to sort of summarize, the glucose metabolism utilization. I think we showed this before, right? Different metabolic pathways and of glucose, and also involves what kind of hormones and how this hormone regulates the different metabolic pathways in fat fasting condition, and how the glucose transporters, GLUT2, GLUT4, contribute to the glucose regulation, glucose metabolism, right? And this is the fasting conditions. So you can just, review all this process. Now let's talk about the diabetes. So diabetes has actually 2 different types, right? Type 1 and type 2. So type 1, the main cause of type 1 is the insulin deficiency, right? The beta cell somehow dysfunction. So the type 1 diabetic patient, the pancreas and beta cell not able to produce enough insulin. So and the treatment is relatively simple. You just supplement with insulin, right? That can cure the disease. And type 2 diabetes is very complicated. The main cause of type 2 diabetes is the insulin resistance and or the decreased insulin actions. So insulin levels not decreased, but sometimes even higher, particularly in the early stage of type 2 diabetes. The insulin levels actually increase, but insulin actions decrease. Right? You have the increased insulin level is just a compensate to the low sensitivity of insulin, right? So insulin and the later stage type 2 diabetes also has the so-called beta cell failure. So the later stage of type 2 diabetes, you also have a lower decreased insulin. So you need insulin supplementation in the later stage of type 2 diabetes. So treatment for type 2 diabetes, we usually encourage them to change the diet and also do exercise. And if still cannot control the blood glucose level, then they can use some anti-diabetic drug to help control blood glucose level. And the very later stage, if they have a lower insulin level because of beta cell failure, then insulin therapy is also needed, right, for the later stage diabetes. And here I just wanted to introduce you some of the the pathological situations in terms of metabolism, right? So here I just wanted to introduce like 3 major metabolic tissues: adipose tissue, muscle, and liver, and see what type of, what kind of metabolic dysfunction, dysregulation occur in the different tissues. So adipose tissue, we know, is a very important tissue that regulates glucose and also lipids, right? So diabetic patients have a problem not only in the glucose metabolism but also lipid metabolism. So so in the adipose tissue, GLUT4 expression level in adipose tissue is decreased. So type 2 diabetic patients have a lower group 4 expression level. Group 4 we know is very important, right? To transport the glucose, in response to the diet, to transport glucose into muscle and edible tissue. And this is a very important mechanism to reduce blood glucose level. So diabetic patients have a lower group 4 expression and also have a decreased translocation the group 4 to the cell surface. So have a lower level group 4 and also lower level of the group 4 on the cell surface. So then uptake of glucose is decreased. So in insulin basically is the most important hormones, right? Can stimulate group 4 translocation to the cell surface. And in the insulin type 2 diabetic condition, insulin resistance, insulin cannot efficiently stimulate group 4 translocated to cell surface. So you have a lower group 4 on the cell surface and decreased group 4 glucose transported from circulation into the cells, right? And so you have a lower glucose level inside of cells, but a high glucose levels in the circulation, right? So this represents the cells, right? This part, and this circulation of plasma. And as I also mentioned, the type 2 diabetic patient also have problems in the lipid metabolism. So we also know that insulin can also regulate lipid metabolism, right? The main function of insulin for terms of lipid metabolism is to inhibit the glycolysis. So if insulin action is decreased or insulin resistance, then this pathway cannot be efficiently inhibited. So you have a higher HSL activity being stimulated, and you have increased the hydrolysis of triglyceride and release cholesterol and fatty acids. So in the type 2 diabetic patient, the fatty acids lipolysis activities increase in the fat tissue, and fatty acids release is also increased. So you see, again, the insulin resistant type 2 diabetic patient have higher levels of fatty acids as well. And we also mentioned before, remember the ketoacidosis, right? The type 2 diabetic patient also have a ketoacidosis because of high levels of fatty acids, and that can promote the ketone body. Sorry, I think we have I don't know, the ketone body. The levels also increase, right, in the type 2 diabetic patients. I don't think the ketone body is in your original slide, right? Just try to add it ketone body. So ketone bodies also increase in the circulation, right? The type 2 diabetic patient. And remember LPL. LPL we emphasize a lot, right? It's also secreted and in response to insulin, and the main function of the LPS to hydrolyze triglyceride in VLDL and chylomicron in circulation and release fatty acids, right? So LPL, because of insulin action is reduced, so LPL secretion, synthesis and secretion is also decreased. So the diabetic patient have lower LPL activities. So that will, what is the consequences of low LPL activities in terms of the triglyceride level? We should have higher, right? So higher triglyceride level, because triglyceride cannot be broken down efficiently by LPL. So you see that in the type 2 diabetic patients, not only have a blood glucose level problem, but also have a lipid, metabolism problem have increased fatty acids, increased triglyceride levels, and also ketone bodies, right? Ketoacidosis. There's something happened in the adipose tissue in diabetic patient, and okay. Now about the muscle. So muscle is very important tissue that uptake glucose, right, a lot because of the the mass, the large mass of a muscle. So so it's really a big deal if something wrong in the muscle, if muscle cannot pick up glucose, and glucose will remain in circulation. So in the skeletal muscle, the group 4 is also the major transporter, right? Remember, group 4 expression is normal, not changing, not different between normal and the diabetic patient. But the problem is the group 4 translocation to the cell surface is decreased for some mechanism, right? So that will causing the decreased uptake of glucose. And also the muscle is also important tissue that oxidize fatty acids, right? So in the diabetic patient, we know the fatty acid levels increase. So, but muscle cannot, pick up and oxidize fatty acids. So that makes things worse, right? So then even causing more increased fatty acids level because of the mitochondrial dysfunction in the muscle in type 2 diabetic patients. So muscle dysfunction in insulin resistant and type 2 diabetic patients also contributed to increased blood glucose levels and increased fatty acids levels, right? And liver. So liver is the important tissue that particularly control the blood glucose level, right? So insulin resistant insulin action in the liver also decreased. And we know that we just mentioned, in the fat condition, right? Insulin levels increase can suppress the glucose output from the liver, remember, right? But in the diabetic patient, because of the decreased insulin action, so this suppression of the glucose output from liver is decreased. So by this kind of mechanism, so you have a decreased glycolysis and increased glycogen breakdown and increased gluconeogenesis and decreased glycogen synthesis. So these two pathways, glycogen breakdown and gluconeogenesis, are increased in the diabetic liver, so that contributes to the increased glucose output, right? So this also contributed to the high blood glucose level in diabetic patients. And in addition to glucose metabolism, liver can pick up a lot of fatty acids, right, from circulation, and makes triglyceride and accumulate in the liver, so causing fatty liver disease. So type 2 diabetic patients also very common, also have fatty liver disease. Right, any questions? Okay, so insulin resistance have some problem with the metabolic inflexibility. So means that the Randle cycle somehow is not working, in the insulin-resistant type 2 diabetic patients. For example, in the lean insulin-sensitive conditions, in the fat conditions, when we have a high glucose levels, and we can stimulate insulin secretion, right? And insulin can efficiently regulate the glucose utilization. So it can efficiently utilize glucose. We can maintain blood glucose level and also lipid metabolism as well. But in the insulin resistant or obesity, in fat condition, glucose increase, insulin definitely increase and even increase more compared to the lean. But because of the action, it's reduced, not efficiently be able to stimulate glucose utilization, right? You cannot, efficiently push glucose into the tissue uptake, right? And also can not be stimulate, be able to stimulate glucose, the utilization. All right, so I think we pretty much finished this session. Now let's move on to the visualization exercise. All right, any questions for the previous session? Questions? No? Okay. All right, so visualization exercise is a little bit different from what we have covered, in response to the normal physiologic condition. So what we need to know in this section, first of all, is what is aerobic and anaerobic exercise. Have you guys heard about this, right? Aerobic and anaerobic exercise. So aerobic exercise basically is a moderate intensity and undertaken for a long duration. So mean that, for example, like mid-distance or long distance, like marathon, this type of exercise considered aerobic exercise, right? And anaerobic exercise usually is high intensity and short-term exercise. For example, like 100 meter sprinting exercise, only takes 10 seconds or less than 20 seconds. This very short-term exercise usually is an anaerobic exercise. The difference between aerobic and anaerobic exercise is utilization of oxygen, right, and oxidative metabolism or glycolytic metabolism. So aerobic metabolism utilize oxygen and is type of oxidative metabolism exercise, and anaerobic exercise usually is glycolytic exercise, mean will not involve oxygen utilization, will not involve the mitochondrial oxidation, right? And exercise intensity. So intensity, usually we use the volume oxygen consumption as a measure of the exercise intensity. So which makes sense, right? We exercise, exercise intensity increase, and we need to use more oxygen to oxidize nutrients and to produce ATP energy. So the VO2 max is a measure of the exercise intensity. And third thing that we need to know that the respiratory quotient. So this is RQ is a way to estimate what kind of metabolic fuel the muscle used, right? So RQ it can tell us. We can base on the RQ, the value of RQ, and to estimate, what type of fuel muscle use. So utilize more fat or utilize more carbohydrate, or something like this. And then the sources of energy fuel for exercise. Exercise, what kind of energy fuel the exercise can use during the exercise. Use, depend on the type of exercise, and also depend on the duration of exercise. And then the fuel utilization in aerobic and anaerobic metabolism. What type of energy fuel can be used during an aerobic or anaerobic exercise. And then the fuel utilization during the post-exercise recovery period, right, is also different from the during the exercise or post-exercise recovery. And muscle adaptation in response to training. So training can change the fuel utilization or fuel metabolism in the muscle. And lastly, we need to know the carbohydrate loading. And this is very important to increase the muscle glycogen store, right, to increase the exercise performance. Okay, so before we talk about this, the specific exercise type of exercise, I think it's very important. We need to know the different muscle fibers, right? How different muscle fibers contribute or involve in the different type of exercise. We have 3 different type of exercise. Type 1, red fibers, and type 2A red fibers, and the type 2X white fibers, right? We have a lot of information in this table, so I just want you to know and try. I'll tell you the way to remember this, right? Probably it's maybe easier. So try to focusing on the type 1 red and the type 2X white. These 2 type of fibers is exactly opposite, in terms of metabolism, right? And type 2A red just in the intermediate. And the second thing you try to remember is the mitochondrial density. And then the other information should be easier to understand or memorize, right? So mitochondrial density. Mitochondria, we know, is very important and that determine the capacity of the muscle fiber, whether the muscle fiber can do oxidative metabolism or glycolytic metabolism, right? Or aerobic versus anaerobic metabolism. So for example, type 1 red fibers have a higher mitochondrial density, have a higher mitochondrial content. And the type 2X white have a lower mitochondrial content. So the color, you can red versus white, actually mostly is depend on the mitochondrial content. If you have a high mitochondrial content, that makes the muscle fiber darker, and lower mitochondria makes lighter. So you can connect this with red fiber versus white fiber. Red fiber usually is a higher oxidative metabolism, right? High oxidative, and 2X is lower oxidative because of mitochondria content is different. So once you remember mitochondrial content, and then the other things that should be easier to remember, for example, the activities for aerobic or anaerobic. Type 1 red fiber, because of have a higher mitochondrial content, so it's able to do the oxidative metabolism, right? So then should be able to do aerobic metabolism, for example, long distance running like marathon. So most of the runners that actually utilize type 1 red fiber during the running to make energy. And then for type 2X white, because of a lower mitochondrial content, so this type fibers were not able to do much aerobic or oxidative metabolism, right? So mostly these fibers do anaerobic metabolism. So usually like short-term, sprinting, usually the type 2 XY fiber is very important for the anaerobic metabolism or short-term running, right? And then we talk about the oxidative capacity, right? So oxidative capacity, again, because of a high mitochondria, so you should have a higher oxidative capacity, right? And low mitochondria and low oxidative capacity, which should be easy to understand. And the glycolytic capacity, because this type of 1 fibers have a high mitochondria, so most of the time they just be able to do oxidative, complete oxidized nutrient, so they have a lower glycolytic capacity. But the type 2X, because of a lower mitochondrial content, so they have a higher glycolytic capacity. Right? Does that make sense? Right? And then the glycogen content. Glycogen content, you know, the high glycolytic, glycogen content is related to the glycolytic capacity, right? Because the related glycolytic capacity and also mitochondrial content. So because the type 2X have a lower mitochondria and higher glycolytic capacity, so that means this type 2X fibers usually heavily rely on the carbohydrate glucose for energy, right? They cannot utilize fatty acids, right? Because lower, mitochondrial content. So that means this type 2X fiber should have a higher glycogen content, right, for their energy needs. So glycogen content should be high in type 2X fibers, and intermediate, of course this type 1 also can utilize glucose as energy, but compared to the type 2X, the relatively low in terms of the glycogen content, right? And triglyceride content, just the opposite, right? The type 2X, because of low mitochondria, so they have a lower oxidative capacity, they have a lower ability to utilize fatty acids, right? So they don't need too much, triglyceride. But type 1 fiber got high oxidative capacity, high mitochondria, so be able to utilize fatty acids. So fatty acids, triglyceride content in type 1 fibers is high, right? Does that make sense? And okay, so in terms of a resistance to fatigue, remember the lactate levels is a major contributor to fatigue. If lactate acid is accumulated, then this is the main cause of the fatigue, right? So this something should be related to the glycolytic capacity, right? So if glycolytic capacity is high, that means this fiber be able to produce more lactate and easy to get fatigue, right? So the type 1 fibers should be resistant. That means not easy to get fatigue. And type 2X fiber easy to get fatigue because of the metabolic characteristics different, right? And that's pretty much all the information I guess you need to know in terms of different type of fibers and the metabolic characteristics, and these two different almost opposite metabolic fibers. All right. So let me see okay. I think we probably just stop there. And I try to make it today is like evaluation day. I know you guys just did the evaluation for the other course this morning. Will you guys do me a favor also evaluate for this course while you're doing the writing, informal writing? And again, I just wanted to tell you guys, and I really enjoyed this class, and you guys did very well so far. So, and for the final, and some of you guys asked me whether you guys need to sign up. You don't need to sign up for December 16th or 20th exam. You just come show up, right? We have reserve this classroom, we have a large capacity to do this. Let's see, so the informal writing. Okay, so informal writing for today, this is the last informal writing. So just describe the difference in the metabolic characteristics of type 1 and type 2X muscle fibers. I just talk about that table so you can try to emphasize your memorization. So focusing on the mitochondrial density, oxidative capacity, glycolysis capacity, and aerobic system used for ATP production, and what types of exercise activity used for, right? So just focusing on this 5 different characteristics of these 2 different fibers. All right, so I really appreciate you guys taking time to do the evaluation. You guys just go ahead and do it. I just pass around the evaluation form.